and welcome to Wildly Curious, where the questions are unhinged, the science is real, and honestly, we're just getting started. I'm Laura.
And I'm Katy. And...
Ever wonder why a bat might glow? Or how frogs made it to space long ago? Dinosaurs trained for delivery runs. We're asking the questions, the weird ones. Wildly curious, where science gets strange and kinda hilarious. For nature's wonders to ancient funk, it's facts and fun rolled into one big chunk. Wildly curious, we're just wonderful. Come join the fuss.
welcome back, guys. What is this? Like how many episodes is this now in? Three?
We're on the second one. Well, third if you count the mini.
3. Yeah. Okay. Well, we're somewhere in the 14th season. Yeah, somewhere. Yeah. And today we're going to talk about mutations and genetics. So it's not going to be super, super heavy. But before that, we want to get into some nature news. And really the big announcement. One, I still don't know if I've won the birding competition. Or when I say I mean we, a team, because it was a team effort.
The Cheeky Warblers.
The Cheeky Warblers, which is an amazing name. I think last time we recorded, it was before that, right?
Yes.
It was before the competition.
Yes.
Yeah.
Yes, just before.
Okay, so very quick recap. Super, super fun. My son and we saw 96 species that day, which is so close to 100, like painful. But we tried to squeeze, find some last minute ones there at the end, and it just wasn't working out for us. But 96 is still
crazy.
A lot.
Yeah.
Yeah. And so I'm not going to give away our total, but we did really, really good. And we are on, unless the other group that won last year saw a ton, we should be pretty darn close because we were right around where they normally. So yeah, so we'll see here probably within the next week or so, or a couple weeks. Yeah, probably within the next couple weeks. So we'll know. But we had fun. I mean, Luke and I had fun. Man, I've never birded that hard. Like, obviously, you guys know if you don't follow me yet on Instagram, Wildly Katy, we did the booby trip. And while that was really fun, and that was like multiple days in a row birding, it wasn't like all day.
Yeah, like a 24-hour marathon. Yeah.
Yeah, because we got up at 2:30, my alarm went off. 2:30, my alarm went off. We went looking for owls, which we heard 2 little screech owl barred owls were like perfect. And then we went to our favorite spot for morning course, which totally paid off. We were going to stay at our first location. Then I was like, called an audible last second. I was like, you know what? We could either trespass and win or go back to the spot where I know like is our favorite spot locally. So we turned around and hurry up and rush back to our favorite spot, which was totally worth it. Because that place alone was like 50-something species.
That's nuts.
Yeah, and so that was so worth it. So that was definitely the right call. And then the rest of the day, like we hit like a lull around like 11. We came home because when I got to let the dog out.
Yeah.
Let the dog out, grab some lunch. And because it's the heat of the day, we took like a 30-minute, I took a 30-minute power nap because Luke had been sleeping in the car on and off. Yeah. And then we went back out in the afternoon. And then we got done around like 9:30 or so. Because by that point, we're like, all right, we already got some owls. Like, we went and found a great horned owl. Yeah, we went and found a great horned owl because where we knew it was going to be. Got that one. And then we were just like, all right, cool, man. And he slept the whole way home. Which, I mean, the last place, because I did one big loop kind of thing. But yeah, so we were right down the road for the last one and came home and that kid was passed out. And then, of course, the next week was the biggest, like the big day. And Luke he was with me again that weekend, and he was like, Mom. I'm like, no, man, I'm still tired from last week. It's okay. We're not going to do it. But it was we had a lot of fun, and I have a fantastic team. It was like maybe like 8:30, 9 o'clock in the morning. And the 2 other ladies sent me a link, and it was a spreadsheet of like all of our stuff. So they're like just as nerdy as insane as I am, being super organized, and like, so it was great. They made it so much easier. They saw a lot too. So we have a really good chance to win. And if we don't, we're going to be neck and neck with the other group. So we'll see, but it was a lot of fun.
We keep having weird birds come up here. I don't know if they're being blown up
from like a southerly wind, but like in the last like 2 or 3 weeks here in Maryland, we've had 15 scarlet ibis.
Yeah. Oh, nice.
A dozen, like 10 Cuban whistling ducks. I just got a call about, and an anhinga. So like Florida is here in Maryland.
Yeah, crazy.
I've seen them before, but like not in Maryland.
Yeah, right. Dang, that's some good ones. I mean, Texas is just odd. Anyway, and 634 plus recognized species is crazy.
Bananas.
And so whenever we do get those random ones that blow into, it's a huge deal because then people are like, let's get more. But yeah. Let's see, your other nature news. David Attenborough is like a bajillion years old.
Happy birthday.
100th birthday. Man.
And he looks amazing.
Yeah,
like,
for
there's
100?
no way you'd guess that guy was 100. I'd be like 80s, no, for sure 80s.
Yeah,
but like...
Yeah. 100?
Truly not 100.
Yeah. Nope. And he's still doing stuff, still recording, still narrating. He's
all
just
that nature.
Right? He's just got the voice. Good. Right? Alrighty. Hang on 1 second, Laura. Let me yell at this kid again. He's turning up the TV. I can hear him. He opened up the door. I'm just like, kid. That's why I could all of a sudden hear it.
Yeah.
Okay. All right. Into it. All right. So we are, like we said earlier, we're going to be talking about mutations and the tiny changes in DNA that can, and how they can create all sorts of just crazy outcomes from just the tiniest little like, blip.
Yeah, literally the tiniest, like so tiny.
One might say microscopic.
Yeah.
So, yeah, just a itty bitty shift. All right, Laura, do you want to give like an overview? So, again, whenever we do these deep dive episodes, guys, it's not like, okay, one disclaimer. You guys got to remember, one, Laura and I aren't geneticists. All right, we are scientists. We are, like our whole background is in science communication things.
We took genetics, but only one. Like, one
thank
course.
God. Thank God.
I
never
loved genetics. It was so cool, but it's complicated.
It is very complicated. Again, genetics is the one that I'm fascinated with, but as long as I'm not graded on it, because it is just like so crazy.
But especially with this stuff where like I can literally imagine like the transcription process and having to like draw the transcription process.
Right.
That's where it gets into the nitty gritty. Yeah.
Yeah. So but we keep it at a very high general level because, again, most of our audience is nature novices. And so we're breaking down really complex sciences so that it's easy to understand. So if we do have somebody out there listening that does know about this stuff way more, cut us a break. Yeah, chime in, let us know. But also cut us a break that it's not going to be deep dive into this stuff because it is going to be just high level.
Yeah, and some of it's hard. Well, and we did a previous genetics episode if you want even more base level. Like we just did genetics 101. You should go back to that one, listen to that one first. This is a little more deep. And like some of this genetic stuff, especially with this, what we're covering today with mutations, I feel like a lot of it is visual.
Yeah, very much so.
So if you're confused and you're able and not driving, pull up a picture because it will help you.
Yeah. Alrighty, well, Laura, then do you want to give like just a very high-level quick overview of DNA
like
and
what a mutation?
Mutations? Yeah.
So mutations are just changes in your DNA sequence or RNA sequence. So mutation, a change. So remember that DNA and RNA, which both are inside of you, is made up of different nucleotides joined together with what we call bases. Those bases are A, G, C, and T. Except if it's an RNA, it's a U instead of a T. So we're going to be throwing some letters around, but those are just base pairs. Those are the things, for those of you that are watching this on YouTube, that are linking your strands of DNA together. So there's two sides and links them up.
They're the rungs of the ladder.
Exactly.
If you're picturing the ladder, you have the DNA, the double helix, this twisty one. It's the rungs
and
of it.
And they always form base pairs of A and T and G and C, except for mutations. But normal, I feel like maybe not normal, but generally that's what they do. So if these nucleotides get jumbled up, or the proteins that make them do, mutations happen, and since DNA and RNA are the blueprints for making proteins that do literally everything in your body, stuff can go wonky. And these mutations can be caused by different circumstances. Could just be a blip in your own code, like all of a sudden your body went rogue for a second. Could be UV radiation. Could be chemicals. Could be, like, there's so many things that can cause mutations. And some of the mutations are beneficial, while others are deadly. So we are going to be talking about there are different types of mutations and different problems that those things can cause.
That's that.
That's that.
All right. Do you want me to go? I can start. So there are different types of mutations, and so Laura and I split them up. I don't know what to call like the groups though. Like there's not names. They're just similar.
Types of mutations?
Yeah, they're types of mutations, but the ones that Laura has versus the ones that I have, they are kind of similar. And I don't know what to call
some similarities.
Yeah, I don't know what to call that bigger group. So you'll figure it out as we go along. So my first one I want to talk about is one of the mutations that basically turns your DNA kind of like into an autocorrect on the fritz. Kind of, but it's frameshift mutations.
Okay.
Right? So normally your DNA is read in groups of 3. So those letters that Laura was saying, it's like comes in groups of 3. Those little 3-letter chunks are called codons. And each codon tells the cell which amino acid to add next when building a protein.
Like some of my stuff happens. Frameshift happens within some of mine too.
Okay, cool.
Like frameshift is like a result of certain problems.
Yes.
Yeah.
So imagine the sentence. Now, Laura said, the letters, but just for simplicity's sake of thinking about it, I'm just going to use all the letters of the alphabet just to make it easier, all right? So think of it if you have a sentence, the cat ate the rat, right?
Oh, I saw this example.
Yeah, everything works. Everyone understands the assignment of, hey, this is a sentence. But if I remove just one letter, so it's like instead of the, it's THC, ATA, TET, HERAT, like it's all of a sudden you're in danger. And that's an extreme example because sometimes these would just be like one little shift, like doesn't really take much. But that jumbling of that shifting just a little bit is basically what a frameshift mutation does.
Which is caused by my problems because I have insertion and deletion.
You do, yep. So a frameshift happens when DNA either gains or loses letters in amounts that are not divisible by 3. Did you hear that thunder? Jeez. So instead of just changing one word in the genetic instructions, it shifts the entire reading frame downstream, essentially. Every codon after that point gets read differently because of the shift a little bit. And this is important because proteins are extremely specific. If you mess up the instructions, the cells can end up building a protein completely wrong, or sometimes it hits a stop signal way too early and that protein ends up like weirdly short and unstable. Story of my life. So what's wild is that these mutations can range from basically harmless to causing major genetic disorders depending on where they happen in that DNA. So a mild example, now each of the ones, I'm going to talk about 3 different types of mutations. I'm going to do a basic overview like I just did. I'm going to give you a mild example, an extreme example, and then some of like the research and importance, just so you guys kind of know where we're going with this.
I feel like we're going to step on each other's. We'll see. Let's see. Keep going.
We'll see. Yeah. All right, so mild example isn't catastrophic whatsoever, which I'm sure we learned about this in genetics, but I totally forgot about it, is blood type. Do you talk about blood type at all?
No, I do a substitution one that deals with blood, but it's not.
Okay,
that.
Perfect. All right, so your blood type can come down to tiny mutations in the ABO gene, and one of the mutations associated with type O blood involves a small deletion that shifts the reading of the frame protein, which, I am sure we learned about that in genetics, totally forgot. So basically, the protein gets cut short and can't finish the job modifying the surface of a red blood cell the way that type A or B blood does, and the result of that is that's type O. Like, that's just what it is. That's just like a normal thing, so not a big deal. While common blood types like A and B are distinguished by simple point mutations, which are single nucleotide substitutions, the creation of the O allele is characterized by a deletion that, like I said, shifts that genetic reading frame, rendering the resulting protein basically non-functional. And that's it. Like, nothing too crazy. You just have a different blood type.
Different type of blood. Actually, arguably more beneficial.
Yeah, it is the universal donor. I'm A negative, and I remember that because I had to get that shot in my butt whenever I was pregnant with Luke because I'm negative.
Being negative sucks.
Yep. One of the, what they say is the alien life, the alien one, the negative. So blood type is a very mild example of a genetic mutation. Now, if you swing it completely the other way, frameshift is Tay-Sachs disease.
I was about to, so I almost did that one, and I was too, so, because I have an insertion and deletion, which I just lumped together because I literally was going to talk about the exact same thing with the frameshift. I'll just do I'll just mention mine in passing, but I didn't do Tay-Sachs because I was too scared to do Tay-Sachs because I didn't want to know too much about it. I was like, it's too scary.
It can be very scary. I do a very high overview. So in some cases of Tay-Sachs, a frameshift mutation affects the hexa gene, HEXA, which contains instructions for building an enzyme called the hexosalinidase A. And that enzyme is basically part of the cell's cleanup crew, for lack of better terms. Its job is to break down certain fatty substances, specifically molecules called GM2 gangliosides, which are inside nerve cells. Normally, your cells are constantly building up things, breaking things down, recycling materials, and there's a non-stop microscopic maintenance operation happening inside your body every single second of every single day. But in Tay-Sachs, the instructions for making that enzyme get scrambled badly enough that the enzyme either doesn't work properly or sometimes barely works at all. So then that causes the fatty substances that are supposed to be cleaned up just start piling up, basically in neurons. And neurons are not exactly cells you want cluttered like a garage, like stuff full of stuff, because it needs that clean pathway and those clean pathways so that you can run literally the instructions through everything. And over time, those fatty substances, as they build up in the brain and spinal cord and progressively damage the nervous system. While most severe and well-known form is infantile Tay-Sachs, what makes it especially heartbreaking is that babies with Tay-Sachs often appear completely normal at birth and can seem completely fine. But then symptoms start appearing within the first couple months, and parents may notice, like, hey, they've hit some developmental milestones, but then they start drastically regressing. So skills the baby had started to learn just absolutely begin disappearing. So it's not like they regress a little bit because they're, newly acquiring a skill and your brain is forming all these synapses to be able to do something. No, it's like drastically starts disappearing. Muscle weakens, develops. Babies may become unusually sensitive to sound, develop vision and hearing loss, seizures. Unfortunately, it's pretty noticeable pretty quick. And as that disease progresses, the nervous system continues to deteriorate. And all that can be traced back to but a mutation that shifted the reading frame of DNA. One tiny change in the genetic letters. And again, you have one example where it's like just changes your blood type, another frame shift, and it's a completely different outcome. Yeah. So, there's honestly, it's still because of the more extreme situations, right? Scientists are so actively studying frameshift mutations to understand them. And they're not,
because sometimes it depends, like frame shifts can be like just a couple or it can be giant sections.
Yep, giant sections. And so now we know so well about how it works. Now they're actively trying to fix them. So it's like, okay, we kind of get how it works. Like you're going to be shifting it. We understand that. But now how can we fix it? And researchers are working on gene editing technologies, like we've talked about CRISPR several times before, and that can actually target specific mutations and tie to DNA. And one major goal with CRISPR is figuring out how to correct frameshift mutations before they cause a severe disease. So it's basically like as we see that this is going to be happening, can we go in there? Because again, if your DNA is always duplicating, can we go in there and hurry up and fix it? And then that way, as it's reproducing and or DNA is duplicating, it fixes itself. And that's crazy, crazy wild, because when you think about what that means, we're talking about like humans learning how to, edit actual instruction manuals of life. And that really has like some positive beneficial outcomes if done responsibly, right? Because anything that we throw at, people can do a lot of harm or it can do a lot of good. So that's just brief overview on the first mutation, which is frameshift.
We'll be back after a quick break.
Katy here from Wildly Curious. Our listeners care deeply about saving wild animals and protecting places they call home. So partnering with Fallow was an easy yes for us. Fallow creates tracking bracelets that let you follow a real animal in the wild while supporting major conservation organizations like the Dian Fossey Gorilla Fund, Cheetah Conservation Fund, and San Diego Wildlife Alliance, just to name a few. To support their work, click on the link in the episode description and use the code WildlyKaty to save 20% on tracking bracelets. Because curiosity is powerful, but action is how we protect the wild.
Yeah, frameshift is really crazy. So that leads directly into mine, which I actually just lumped two of mine together because insertion and deletion is really just it's the flip side of the coin.
Yeah.
So the outcomes might be different depending on what's been studied, but in general, insertion and deletion typically just leads to frameshift. But basically, insertion is a base pair is added where it doesn't belong, or insertion is when you add in things that don't belong, and deletion is when you delete when things are deleted from the sequence. So like Katy was saying, if it's just a single base pair that's added in, that causes a frameshift. Or if it's two that are added in, it causes a frameshift. If three are added in, like Katy was saying, if it's divisible by 3, 6, 9, if like a group of those are added in, it doesn't cause a frameshift, but it can create just an entirely new amino acid. So throwback to biology and genetics of when DNA is being read, it's being read in sets of threes. Every set of 3 creates an amino acid. Lots of amino acids create a protein.
Yep.
Proteins build who we are. Okay, so like build. So if an entirely new amino acid is being created, I mean, sometimes it's good and sometimes it's bad.
Yeah.
So what are the outcomes? One, it can totally jack up the amino acids being formed, which can change the subsequent protein that's being read, especially if it causes translation to stop early. Translation is what happens. There are little things on your DNA transcribing it. I like to think about it as like little tiny people with typewriters.
Yeah.
And all the little tapes just coming off.
Yeah.
And it's creating new things. They're just translating your DNA and making lots and lots of proteins. So what we want to happen is that process to just not stop, at least in chunks, to make these proteins. But if something is wonky, let's say these little guys who are reading your DNA, they're like, this doesn't make any sense. Forget this. I'm not reading anymore. They just stop. So then you have a really jacked up protein because it's not as long as it's supposed to be. I want to give some examples of, a good a lot of times people talk about mutations as being inherently bad. And what we've said is that it doesn't necessarily have to be.
Yeah.
So an example, a beneficial, because there are beneficial, the entire human race was made because of mutations, right? Like if you subscribe to any form of evolution, we got here through mutation.
Yeah.
Or
adaptations of anything. Like let's just say, even if you don't believe at all in evolution, don't
form of change.
Yeah. Is mutation. 100% this. Yeah.
Yeah. But more specifically, I found a cool one, which is a 32 base pair deletion in a specific gene known as CCR5. It removes one of the receptors from a white blood cell, which makes you basically immune to HIV.
Interesting.
Yeah. So that'd be a great mutation to have.
Yeah.
You just can't get HIV.
Yeah.
And that's one.
Are they doing anything with that to make
I didn't get a chance to look into it.
Okay.
I totally should have. I just looked at it, literally just looked it up. Because I was like, oh, I should have talked more about beneficial stuff. Not just the doom and gloom.
Yeah.
But surely, right? Because I guess you'd have to do some gene splicing here. I mean, like, right, the future is gene splicing. But comes with inherent risks. I was just reading an article that they found the first ever case of a brain tumor caused by gene splicing, which is so like, but it might save your life. And the brain tumor they removed, the person was fine. It's just like, yes, messing with your body can cause cancer.
Yeah.
Definitely. So
well, was it cancer or was it just was it just a benign tumor?
Oh, yeah, you're right. I guess it was just like cancerous growth.
I mean, it could be. Yeah.
Yeah. I didn't read enough. But yeah, so whether or not it was benign or not, messing with genes can cause issues. But if we could figure out how to harness it, we could do some crazy cool things.
Yeah.
One, though, that's not so beneficial, but like on a scale of like, I chose to focus on ones that didn't cause death.
Yeah.
Because I just didn't want to read too much about it. It was too depressing.
Yeah.
Lots of genetic issues have their own help groups and own support groups and like, you know, advocacy groups. You should totally look it up. But this one I wanted to talk about is the deletion of part of paternal chromosome 15. So remember, you get 2 chromosomes, one from your dad, one from your mom. This is a problem with your dad's number 15. It's Prader-Willi syndrome. It's spelled Willy, but it's Villi.
Yeah.
And typically, so 60% of the time, it's caused by a deletion. There are other because mutations can happen for lots of reasons.
Yeah.
This can happen, but deletion is usually the problem. And basically what happens is it makes your hypothalamus not work correctly. And your hypothalamus does a lot of things in your brain.
Yeah.
As infants, it causes poor muscle tone, poor sucking reflex, and distinct facial features. So like just like a hard time growing and thriving. And then as they get older, there's a lot of symptoms, but some of them are delayed puberty, poor growth, behavior issues, cognitive issues, and the worst one, I think, is being hungry all the time.
That would be horrible.
So imagine the hell of never being full.
Oh,
I mean, truly, they literally lack the ability to be full.
But I mean, okay, just mentally, the strain and stress that would put on, you know what I mean?
You'd always be thinking of food.
Yeah, man.
Because, like, when I'm hungry, like Katy knows, like, I'm eating every 2 hours and I'm hangry. Like,
Laura gets hangry. Yeah, Laura definitely gets angry.
Hangry 24/7. I bet they're different people. Like, can you imagine the personality shift if they were full?
Yeah.
So what happens is they crave food all the time. Not only do they crave food all the time, but they actually have a lower metabolic rate. Both of which can lead to severe obesity.
Yeah.
And all the issues that come with that. So, like, I mean, these people have to go through serious therapy to learn how to control cravings. When they're little, people have to, like, lock food up. They've been known to hoard food, eat frozen food, and even eat garbage because they're so freaking hungry.
Yeah. Well, their brain is telling them that they're hungry. Like, their stomach is physically
They're not
full,
actually
but
starving. Yes. They just can't. They never feel satiated. So although it doesn't cause death, there are serious complications of, one, obesity and, like, mental toll,
it's
but
almost
also
like
sterility and osteoporosis.
It's almost like a secondary, like death is like a secondary because of how much of the symptoms, it's a secondary cause of it, you know?
Yeah. And so it's thought to be between 1 in 10,000 to 1 in 30,000 births. So thankfully not a ton, but man, it would be horrible. My thoughts go out to anybody that has it.
Yeah, that would be a tough one.
And then on the flip side, insertion. So in this gene called the FMR1 gene, which is located specifically on the X chromosome, which is the one you get from your mom, and you get one from your dad. If you're a girl, you get 2. If you're a girl, you get 1 if you're a guy. There's an expanded CGG triplet. So those 3 little base pairs, CGG, it happens multiple times. And that causes Fragile X syndrome.
Yeah. I remember a lot of these. Once you say them, I'm like,
me too.
Oh yeah. I don't want to look too excited, but any of my facial expressions is like, hey, I remember something from that horrible
Recognize
class
that term.
that we
Fragile X syndrome basically turns off or silences the FMR1 gene, which is responsible for creating these special proteins that build nerve synapses, so the things that go between nerves. And because of this insertion, it makes the X chromosome look all wonky and look broken or really fragile. And so the symptoms of this are intellectual disability, anxiety, depression, and OCD, behavioral issues, and specific physical traits. These people are at a higher risk for many health issues, including dementia, migraines, chronic pain, and seizures. So all problems with like nerves because of nerve synapses. And unfortunately, it is a dominant trait. So it's, if you've got it on an X, you're going to have it, and it's sex linked. So that means that if you're a dude, you're way more likely to have it than a girl, because as soon as you get one X, that's it. But even if you are a girl, if you've got even just one broken X, you'll have it. So it's about 1 in 7,000 males and 1 in 11,000 females. Apparently, it's like one of the most common reasons for genetic intellectual disabilities. So beneficial insertions and deletions happen, but there's also some really terrible things, and even worse than this. But
again, it goes back to just how wild it is of like, so for our viewers on YouTube, you will see I have a new shirt on. It's because I was getting my son his lasagna and I flopped it out on the plate and it just
splattered.
New shirt.
So
okay.
stains too.
Right? Yep. All over my shirt. Anyway, we are good. Okay, so what I was going to say was that, yes, it's like, it's horrible, but at the same time, when things go wrong, it can go very, very wrong. But at the same time, you got to remember, our bodies are doing this a bajillion times.
Yeah.
And so it's insanely impressive.
That it's not constantly happening. And that it could be constantly happening, but that the so wrong is so rare.
Yeah. Yeah.
Like, I think it's important to keep that in perspective.
Yes, that like, hey, this is going on thousands of times, and it's insane that our bodies can even get it right this many times all the time at all, because of the process. So that is really, really, it makes you appreciate the process that you literally don't see. And we didn't know for thousands of years. We had no idea it even existed.
Yeah, it's literally like straight up magic, or like it is crazy when you really think about it. The fact that like, we're not just single-celled organisms here. We are making billions and trillions of proteins and like
decisions a day that we're not even aware of.
Most of the time, it's just right. It's just right.
Which is
just
crazy.
Happens. Yeah.
Alrighty. So my next one then is duplication. All right.
Okay.
So this one I always got to start with an example. That's just how my brain works. But it's DNA's equivalent of hitting copy and paste too many times. All right.
Which is kind of like insertion.
Yeah.
Or like.
Yep. Yeah. A duplication mutation happens when a section of DNA gets copied more than once, exactly what it sounds like. Sometimes it's a tiny section, sometimes it's an entire gene. Sometimes it can even involve huge chunks of chromosomes getting repeated. So it could be a variety of different things. And unlike a frameshift mutation, where the instructions kind of get all scrambled up, duplication mutation still keeps the original instruction. So the cat ate the rat. It's just the cat ate the rat, the cat ate the rat, the cat ate the rat. More so when it should just be one sentence. Now it's like 3 or 4. So, I mean, obviously, like, it sounds, for the most part, harmless, right? Like, if one cookie is good, obviously 12 cookies are better situation.
Yeah.
But unfortunately, that's not always the case, because cells, just like anything in biology, operates in a balance, right? Proteins need to be produced in very specific amounts. Too little can cause problems. Too much can cause completely different problems, just like in an ecosystem. Too many of one species, problem. Too many, too little. It all about balance. So duplication mutations can create all sorts of odd outcomes depending on what gets copied and how many extra copies are actually there. Some of them can be surprisingly helpful. One of the examples that I do remember this from our genetics class was the AMY1 gene, AMY1, the number gene. And this gene helps produce the salivary amylase, which is the enzyme in our saliva that starts to break down starches while we're still chewing food. So before your food even reaches your stomach, our body is already going, excellent, potatoes, like let us begin. And that's my head, like
Let's begin.
like, because again, my brain, and I have to use potatoes because that's by far my favorite starch, but I'm also German. And so there's just something in me that I'm like, oh my God, potatoes. Give me any form of potato and I'm a happy
I feel like most
camper.
of us are like that. Yeah,
Oh
100%.
God.
Pickles and potatoes.
I forgot, though, whenever I was relooking all this up, that humans, not all of us have the same numbers of copies of this gene, right? Some people have more copies than others. Yeah, because duplication mutations over time created extra versions of the gene. And populations historically like starch-heavy diets, like Laura and I were like, oh, I feel like that's everybody. Honestly, down here, people eat potatoes, but it's not like a staple of the diet because, again, where I'm originally from in Pittsburgh, it's cold. It's very much so like German, Polish. You know, we have our pierogies, we have our lactose, we have the very filling foods because it's cold and your body needs to stay warm. It needs that extra energy. We're down here in Texas because it's so dang hot. If you're eating foods that make you feel heavy, you just feel miserable because it's 120 freaking degrees in the summer. So you eat tacos that are lightweight, like the light filling, so you don't need as many. So this heavy starch diets, things like rice, wheat, potatoes, those populations tend to have more copies of the AMY1 mutation, which just means that their bodies
just thinking about it.
Potatoes, man. I'm a potato and a pickle girl, like through and through.
The most Pittsburgh thing ever. Pickles and potatoes.
Dude, I can't. I have Pittsburgh blood in me.
I haven't met a form of potato I don't like.
Oh, me either. I can make a podcast episode just on potatoes.
On just potatoes.
On just potatoes and all their functions. I would be so happy. We'll dog ear that
that'll be our
episode
next Thanksgiving one.
Yeah, just
Instead
potatoes.
of turkeys and pumpkins, we'll just do potatoes.
Potatoes, yeah. So sometimes though, evolution basically is just like, hey, this is actually beneficial. You have a food, this population, especially, in Europe, wherever it's cold, these groups are like, hey, these starchy foods actually do us well. It helps us, you got to have food and stay warm. And so the body's kind of started shifting over time and provided more of that, like the salivatory protein in your saliva to help break it down. And so it's wildly beneficial for groups. I mean, that can be beneficial. One of the more extreme examples, though, where duplication can go very, very wrong is called Charcot-Marie-Tooth disease type 1A, usually shortened to CMT1A, because that everything else is just a mouthful to say. But this condition is commonly caused by duplication of a gene called PMP22. And PMP22 helps maintain myelin, which is the protective coating wrapped around nerves. So think of myelin kind of like the insulation wrapped around electrical wiring, right? It helps the nerve signal and travel quickly and efficiently throughout the body. But, there are also... This is kind of one of those examples where duplication mutations kind of get sneaky because you think, oh, extra copies of like a helpful gene could make would it make it work even better? And unfortunately, no, because again, biology is built entirely around the concept of like too much of a good thing isn't always a good thing, if that makes sense.
It has to be just right. It's your body is Goldilocks.
100%. So people with CMT1A have an extra copy of the PMP22 gene, which leads, like we said, to too much of that protein being produced. And instead of helping nerves, that imbalance actually disrupts the myelin and damages nerve signaling over time. So people with CMT1A can experience muscle weakness, loss of sensation in the hands and feet, balance problems, difficulty walking, and so on. And so again, like this can just be like one extra copy, and that's what causes this. to me, again, the duplication one, just like all of them, it's just crazy how one little duplication in one spot, same thing as the last one I talked about, it causes like something very mild, like, okay, you salivate, more efficient for your starch food. And then this type of duplication, like, is too much of a coating, and it breaks down your nerves, essentially, or it prohibits them from communicating as effectively. And so scientists, they're definitely studying the duplication mutations intensely because they play such a huge role in human disease development, the evolution, and cancer research. So they're really right now trying to understand something called gene dosage, which is basically the question of how many copies of a gene is too much. Because again, sometimes that duplication is like, okay, like, again, 3 cookies is okay. 12, too much. But like, how much is too much? Is it 12 or is it 6?
It all depends on how much it's bending your protein. Like, it makes a certain shape. It's like, it's basically creating an origami. If you add in too many folds or not enough folds, it doesn't make the animal you're trying to make.
Exactly. And so that's kind of what scientists are really working on right now with that. You know, and again, using a lot of the same sort of technologies and studying this, because again, a little bit is okay, too much is too much, and just trying to find that line. And then, hey, can we fix, can we reverse, can we cut this out? If and like, where is it going to be too much? So if 12 cookies is too many, if we backed it down to 6, like, is that okay? Like, is that an okay? Can your body function with some of these conditions that you end up getting from duplication? So yeah, that's all I have for my second one.
Interesting. So just my other one, and the most common form of mutation, I was going to start with this one, but insertion and deletion just added so well into yours that I was like, I just got to continue it on because that's frameshift. But the most common type of mutation is substitution. So this one, it's just a flippity flop, a switchy switch. It's just when one base pair is replaced by another one. Although there are 2 types of substitutions. There's a transition. So that is when, hold on, I got to get this right. Okay, so you know how I said that there's these 4 letters, A, G, C, T. And I said that A and T always go together, and G and C always go together. Now a transition, and like, okay, imagine 2 of them are always on the top and 2 of them are always on the bottom. A transition is when the same 2 on the top switch with each other. Okay, so that would be an A and a G flip or a T and a C flip. So it's just transition from one spot to the other. Then there's transversion, which is when they completely flip sides. So when an A and a G pair up, which they don't ever do, or a T and a C pair up, and they never do. So 2 different types of substitutions causing different issues, but either switching with your partner side by side, do-si-do, or cross the room. So
again, it's just like, it's so
of your genes.
But it's so, again, it goes back to, it's so mind-boggling to me that we're not just like goop mush. Like the fact that for the most part, humans, we are evolved as we are to do what we can do, and that this process has to work perfectly every single time, all the time, is just, it's nuts to me.
Yeah. I mean, I think that's where some of like my faith comes in, because I'm like,
craziness.
Perfection.
Yeah. Someone's got OCD. Like,
so the outcomes, we've also failed to mention, I think, so there's beneficial mutations. There's the bad, when it goes bad, but there's also just silent mutations. So sometimes the outcome is just nothing. It's just nothing.
Yeah.
It's literally the same outcome before the switch ever happened. Sometimes it just works out that it's the exact same thing. The person reading it was like, they didn't get messed up. They just kept reading and made the same end product. Kind of like, sometimes you see those sentences online where you're like supposed to read it and you didn't notice that one word was wrong.
Welcome to freaking ADHD.
Right.
Yeah.
Like,
my
you
brain
can just run with it and it doesn't matter.
Yeah. My brain doesn't even catch most of that because it's just filling in the blanks. It's like, oh, it just assumes that this is how it's supposed to be. Yeah.
I make a lot of assumptions too when reading directions and things like that. Like, I'm like, yep, got it.
Yep. Yeah.
Same outcome, usually. And then so that's when nothing happens. And then there's a missense mutation, which is when new code is created that can impact formation. So that's when these frame shifts that's when what is read off is changed.
Okay.
But most of it's still there. Like,
Okay.
it's just all jumbled up.
Okay.
And then there's nonsense mutations, which is stopping translation. That's when the reader is like, I literally cannot.
done.
This makes
Yeah.
no sense at all.
Yeah.
I'm done.
Yeah.
So they stop translating prematurely, which can have major impacts, and because the protein's just not even there.
Yeah.
Time where it's beneficial that a substitution happens, probably my favorite example, is lactase persistence, which allows us to keep drinking milk. And if there's one thing I love almost definitely as much as potatoes, it's milk.
See, I've gotten away, and again, I think it's like a heat thing. I've gotten away from milk. Cheese, however, that's
you're like a no going nowhere. Yeah. Bring me all of the dairy. When I went dairy free for 1 month for the baby, just to see if it would make any difference, I was like, I never realized that this is probably my biggest food group. Cheese isn't everything, and milk isn't everything.
Yeah,
I miss a good glass of milk. So lactase persistence, there had like over time, like humans weren't always able to drink milk forever because all the other animals cut it off after infancy, and then you become lactose intolerant. So like those of us that still have lactase persistence, that's all because of some substitutions that happen, a whole bunch of them. So like single base pairs swapped with another one, usually most prominent in Europe, probably again because of like, I don't know, dairy calories, putting on weight, needing to be, because it's cold.
Yeah.
Who knows? But a beneficial mutation that allowed us to keep eating dairy. But another one that is both good and bad, because sometimes mutations can be both, is sickle cell anemia.
Ooh, yeah.
So that is when the A and the T get flipped in the sequence that codes for hemoglobin, which is what carries the oxygen in your blood, and a completely new amino acid happens. So this is a missense mutation. So it's a mixed up one, but it still works. So this impacts the way the protein folds and the overall shape of the blood cell. They're supposed to be round when they're folded. Blood cells are supposed to be round all the time, whether they have oxygen in them or not. But when this particular blood cell with this mutation becomes deprived of oxygen, they clump together and they become sickle shaped. So like those crescent moons, which can interrupt blood flow and cause pain and fatigue. But on the flip side, it does make you mostly immune to malaria.
Yeah.
So win, not win. I mean, you can be in pain but not get malaria. So depending on where in the world you live,
isn't this
depends
one
on how good.
African Americans?
It's more prevalent in African Americans.
Yeah.
Which also, I mean, like depending to like, it probably was allowed to persist because there are some benefits to this. One, it doesn't it's not necessarily fatal.
Yeah.
Although it can disrupt blood flow. Okay.
Yeah.
So it can eventually become fatal.
Yeah.
But if
but again, is that the actual?
There's lots of malaria.
Yeah. But is that fatal because of the actual mutation, or again, is that like fatal from secondary? You know what I mean? Like the secondary
probably,
causes of it.
I don't know. I think it can cause clots. But if you're making it to reproductive age, you're able to pass this along. And then if you're immune to malaria, you're more likely to be able to pass on your genes. So like this has persisted because all of these mutations have persisted because they either happen randomly or because we've never gotten rid of them because they're not too detrimental, like the lactase thing.
So I just Googled it, and yes, it is. I knew it was serious, but it is life-threatening because of one thing that you didn't mention is it was because it gets sticky, like the blood.
Oh, right.
Like
they clump together.
They clump together,
and
that is more so the problem than anything, which can block blood flow, leading to fatal complications, acute chest, anything with your heart, strokes, organ, kidney failure, severe infections, and those kind of things. So it is because of the stickiness. Yeah.
Yeah. So like a clotting problem and blood flow.
Blood flow. Yeah.
So yeah, that substitution, anything from being able to eat and drink dairy to sickle cell anemia.
Yeah. Well, guys, I'm going to wrap it up then with inversion mutation, our last one real quick. These are honestly one of the weirdest ones to visualize, at least for me, because the DNA itself doesn't necessarily disappear. It just flips it backwards.
Oh, okay.
So your chromosomes can break and rotate a section of DNA like 80 degrees and then like jam it back into place if
Reattach.
it needs to. Yeah.
The little workers are like, put it back.
Yeah.
And they just didn't realize that they did it wrong.
Yeah. Oh no. So imagine, yeah, right. So imagine if you had a DNA sequence that originally looked like A, B, C, D, E, F, G. An inversion would be A, B, E, D, C, F, G. So it just kind of like flip-flops a section of it. Could be bigger or smaller. And so again, it's like the information is still technically there, but now part of it is reversed. And depending on where that inversion happens, the effects can range from like something that's not noticeable at all to severe genetic diseases. And because genes don't just depend on the information itself, they also depend on order, orientation, timing, and regulation. Once again, biology is an organizational nightmare. It should be, but it's not. It's just wild again. So one famous example is something called 17q21.31 inversion polymorphism. So that inversion occurs on chromosome 17. And so many carriers have no obvious symptoms whatsoever. So there's no consequences to this, no sudden urge to really know what's going on. And scientists think some of the inversions like this may actually stick around in populations because they help preserve useful combinations of genes that actually end up working pretty well together. Which is kind of interesting because it means something like evolution, like we talked about, sees a chunk of DNA flip backwards and it was like, well, that works. Let's just leave it there. Yeah, let's just leave it there because it doesn't really do anything. So most people go through life having no idea they even have this with no symptoms until they get genetic testing, but probably for something completely different. And then they're like, oh, hey, by the way. And that's like with me. So I have an autoimmune condition and the whole MTFR thing, how my body doesn't break down everything as well as it should. I had no idea that was even that was even a problem for me until I started getting some genetics testing done to figure out what the heck, like, why did Katy almost die? Like, why is her body trying to kill her out of nowhere? And so whenever they started doing all the testing for that, that's when I started seeing some underlying things that I never would have known until I went through all that testing. One of the more extreme examples, though, of inversion is, you already talked about blood, so hemophilia A, so hemo, blood, is a genetic disorder where the blood has trouble forming blood clots properly, so you can imagine...
The opposite of sickle cell.
Yep. Yeah. So you can imagine how the problems that could lead for that. And so...
Yeah, that was like a big problem with the royal families in Europe, right? Hemophilia.
Yeah, and it definitely is seen a lot more in some cultures than others. So the F8 gene contains instructions for making clotting factors, which is, in this case, it's the factor VII, the 8, which is one of the major proteins your body uses to stop bleeding. So basically, when you get injured, your body launches this insanely complex biological chain reaction involving clotting factors working together to form a stable clot. And your blood is basically running emergency repair constantly. But whenever you have that inversion mutation, part of the F8 gene gets flipped and it just is around backwards. And so even though the DNA part is still technically there, the instructions become disrupted badly enough that the body can't properly make a functioning factor VII, the F8, like it should. And without enough factor VII, blood doesn't clot the way it should. So people with severe hemophilia A may experience prolonged bleeding, easily bruising, internal bleeding into muscles and joints, chronic joint damage over time. Yeah. So, I mean, obviously if your body, and again, it's not, people think, okay, if I get a scratch, like that's the only time it would bother me. No, like our body is internally like constantly forming clots that we don't even know about.
Is like, yeah.
And so again, it's just that's such something that could be so detrimental is just caused by a section flip backwards. Yeah. And scientists are interested in inversion mutations right now because for a long time, they were really difficult to detect. Like they knew something was wrong, but they couldn't really figure out what because they would be reading through the chain. And they're like, well, all the pieces and parts seem to be there. What the heck is going on? And so older genetic technologies were good at spotting missing DNA or extra DNA, But inversions are a lot sneakier because that DNA is still present. So even some of the older technology, even if it was in order, the machines were like, well, it's all still there, guys. And they're like, yeah, no, it's not. It is there, but it's all out of order. So with a lot of the newer long-read sequencing technologies, researchers are discovering that inversions may play a much, much bigger role in a lot of these mutations than we originally thought. And so now because of the newer technology and they're easier to detect, just like everything else, a lot more research has gone into it to try to figure out, hey, how can we prevent and fix these things?
Yeah, so mutations, the good, the bad, the ugly, some are good, some don't do anything, some are bad, and some are real bad.
Yeah.
But like we were saying earlier, it's kind of just remember your body's doing this all the time, and for the most part, it's good to go.
Yeah, which is crazy in so many aspects. So, all right, guys. Well, next week we have another short episode. Until then, visit us on Patreon. I have been posting more content on there. Support us if you can. It helps keep all of this up and running because it costs monies. So any little bit helps there. And yeah, like I said, next week it'll be another episode of I bet you never heard of that animal or whatever we're calling that series.
Yeah.
Something along those lines.
You probably never heard of me.
There we go. Yeah. You probably never heard of it. Not betcha. All right. So until next week, guys.
See ya.
Ever wonder why a bat might glow? Or how frogs made it to space long ago? Dinosaurs trained for delivery runs. We're asking the questions, the weird ones. Why?